Enzymes are really important proteins that speed up the rates of reaction, such as in photosynthesis, respiration, and protein synthesis. Enzymes are known as biological catalysts because they lower the activation energy of the reaction, hence speeding it up. They are unchanged at the end of the reaction and can be reused. Enzymes can work in different ways. Some enzymes help to break down large molecules into smaller ones, others build up large molecules from small ones, and some enzymes help change one molecule into another. So how do enzymes work? Enzymes work in conjunction with substrates. The enzymes and substrates are always moving, and occasionally they collide at the right speed and orientation so that the substrate fits into the enzyme at the active site. Collision theory dictates that collision must occur with sufficient energy and in a specific orientation for a reaction to occur. Enzymes are specialized. Their active site matches the shape of a specific substrate that they can react with. The enzymes and the substrates fit together using a lock and key mechanism. Once the substrate is in the active site, the reaction takes place. The required product is produced, and the enzyme releases itself and carries on moving around. In this example, the enzyme is breaking down the substrate into two smaller products. It could be protease, which breaks down proteins into amino acids, or carbohydrase, which breaks down carbohydrates into glucose, or lipase, which breaks down fats into fatty acids and glycerols. Hydrogen peroxide is often formed as a result of the reactions in cells, and if it is left behind to build up, it is harmful. Luckily, we have catalase enzymes that are really fast. They break the hydrogen peroxide down into harmless water and oxygen. Enzymes can help build up molecules like this, but the process is still exactly the same. Whilst enzymes do fantastic things, they are sensitive. Each enzyme has optimum conditions under which it works best. Firstly, there needs to be enough substrate around. They need a high enough substrate concentration for the reaction that they catalyze. If there is too little substrate, then the rate of reaction is slowed. Sometimes, if there is too much product around, then the reaction slows because the enzymes and substrates have less chance of bumping into each other. So, the product needs to be removed for a higher rate of reaction. Enzymes also have optimum pH and temperature conditions. Up to a point, an increase in temperature causes increased rates of reaction because there is more heat energy. More energy means more collisions. However, above a certain temperature, the rate drops off due to denaturation. We will look at the effect of pH and temperature on enzymes in our video Denaturation of Enzymes. The optimum pH and temperature conditions are specific to the condition in which they work in. An enzyme that works in the stomach, for example, would have a more acidic optimum pH. And of course, there need to be enough enzymes around for the rate of reaction to be optimized. So we know that enzymes and substrates fit together at the active site to form a lock and key mechanism. The enzyme then releases the product and can be used again. They are sensitive to temperature and pH, and there needs to be a sufficient enzyme and substrate concentration for reactions to occur. Enzymes not only control all kinds of reactions such as in photosynthesis, respiration, digestion, and protein synthesis, but we also make use of them in day-to-day life. Protease and lipase enzymes are used in biological washing powders to remove proteins and fats from stains in our clothes. We also use enzymes in our food and drink industries. Pectinase is used to break down the cells in fruit when making juice so that more juice is released.